Hello and welcome to episode 2. This is our first interview of the show, and we are starting off with a bang with some very exciting science that could have huge implications for neuron healing and regeneration. This is an interview with Dr. Michael Levin of Tufts University, and I'll give his bio here in a moment. But to give you a little teaser of some of the science you're about to hear about, he and his lab have figured out how to amputate the hind limb of frogs, adult frogs, and then apply bioelectric signals to then regrow that limb. It takes, I think, over 12 months, but they regrow a fully functional, fully innervated hind limb in these frogs. And for those who don't know, frogs are like humans. You cut off a leg, it's not going to naturally grow back like in a Planarian or other salamander models. And another experiment that we're going to talk about is how they have figured out how to regenerate severely damaged brains in tadpoles, and in two different experiments, one where the damage was caused by teratogens, carcinogens, teratogens like nicotine, those kind of things, and then another group being damaged by an actual genetic mutation of the Notch gene. And in both cases, it's not just that they're regrowing these brains and they're still highly, affected and dysfunctional. They're actually indecipherable from healthy controls, which is amazing. And then to become even, more intriguing is that with the group with the Notch mutation, that genetic mutation persisted. They didn't change that. They just applied the bioelectric strategies. And while it had this genetic mutation that caused the initial brain damage, somehow it was able to get around that issue and regrow a brain indecipherable from healthy control. So really, really cool, exciting stuff that you're going to be learning about and hearing about today. Now, I've recorded a lot of these episodes before we actually launched the show, and I recorded this one probably about a year ago. And I'm pleased to report that in the meantime, Dr. Levin has graciously helped to design some experiments with a neuromuscular researcher at a different university to apply these bioelectric strategies to mouse models of spinal muscular atrophy, ALS, and nerve crush injury, and that we might be able to include spinal cord injury as well. That's yet to be determined. But those experiments should begin later this year, 2024. If you want to keep up with those, as well as any other news, updates, recent published papers on neural regeneration, we are going to be sending out a weekly email. So you can go to neuronregeneration.com and sign up for our email newsletter, and you'll get that in your inbox every week to stay current on all of these things surrounding the topic of neural regeneration and healing. So before we dive in, here is a quick bio on Dr. Levin. Michael Levin is a Vannevar Bush Distinguished Professor and Director of the Allen Discovery Center at Tufts University and Tufts Center for Regenerative and Developmental Biology. He received dual bachelor's degrees in computer science and biology from Tufts University and a PhD in genetics from Harvard University. His postdoctoral training was in the cell biology department of Harvard School of Medicine. He has published more than 350 papers and is also an associate member of the Weiss Institute of Harvard Medical School. Okay, thank you again for joining us. And now please enjoy my conversation with Dr. Michael Levin. All right, Dr. Levin, thank you so much. I've been so excited to be able to have this conversation with you, and welcome to the Neuron Regeneration podcast.
Well, thank you so much. Happy to be here.
Absolutely. So I wanted to just start out with a couple of quotes from some of your past interviews to set the stage a little bit and kind of
Mm.
cue you up on the first thing that I wanted to cover. And the first quote was that you had said, disciplines are not real. That's just one lens. That they're all using one lens to look at things, and they love that lens. And all the things that could have been done with a single lens has pretty much been done. All the easy things that could have been solved by a single perspective, I think, are mostly solved. And everything else needs you to crack out of that lens and use multiple approaches that are informed by different ways of looking at the world. I love that quote because, and it's very much why I'm doing what I'm doing and the way I am, and trying to get people from different disciplines to talk across disciplines, types of trauma, types of nerve damage, as well as various neurodegeneration, because there have been such huge long-term challenges, across the topic of neuron repair and regeneration. So it's exciting to come across someone doing work that really is approaching it from a different angle than, say, some of the things like gene editing or modulation or stem cells or any of the various things that are being worked on, which certainly show a lot of promise and already have had major breakthroughs. But I'm really excited to kind of bring what you're doing and your work to this topic today. You use the analogy of computer hardware versus software to explain those different therapeutic approaches versus, like, say, what you're doing in bioelectricity. So, can you start by summarizing bioelectricity and then explain that analogy to give the viewers and listeners some foundation of what we're going to be discussing today and what you're working on?
Yeah. So let's see. People know bioelectricity best, perhaps, as the modality that enables individual neurons in your brain to work together to give rise to an emergent, coherent individual. So you have goals and preferences and memories and other cognitive aspects that don't belong to any of the neurons. They belong to you as an emergent, higher level entity. And so bioelectricity is that kind of cognitive glue that binds the actions of these competent individual cells into something that's more than its parts. And this is literally electricity. So the thing that allows information storage, processing, integration, all of that in your brain is the ability of the individual cells to form networks and talk to each other electrically, and that's what binds them together. Now, one can ask where did the brain learn this trick? Because it's a very cool trick, and the reality is that evolution discovered this trick a long time ago, long before nerves and muscle came on the scene. So it's not just for behavior in three-dimensional space, which is where we mostly study it, but it's also back from around the time of bacterial biofilms. These microbes were already using electrical networks to coordinate their activity towards larger scale goals. And so, what my lab is really interested in, and several other labs throughout the world, is this idea that you can actually use a lot of the tools from neuroscience, from behavioral cognitive science, and so on, to try to understand how evolution pivoted these various solutions across the the tree of life. So you have the most primitive organisms are mostly dealing with very tiny little metabolic goals and physiological goals, and then eventually you get multicellular creatures, and those cells need to work together on very large goals. So building whole organs and regenerating limbs and suppressing cancer and keeping to a particular target morphology, a shape, particular shape of the body. And then eventually evolution pivoted that into so what it had was using these electrical networks to navigate what we call anatomical morphospace. So this is the virtual space of all possible anatomical configurations for a given body. And so the embryo navigates that space, goes from a single cell all the way to the shape of whatever it is. And then evolution pivoted to some of those same strategies by speed optimizing the process. So neurons, of course, do this much faster than other cell types, and used it to navigate three-dimensional space. So whereas now the electrical networks in your brain control muscles to move you through three-dimensional space, the evolutionary origin of all of this is the use of these electrical networks to process information that tells individual cells what to do in terms of differentiation, proliferation, and so on, to move the configuration of your body through this virtual morphospace, this the shape of all possible anatomical configurations. So very much like bioelectricity in your brain provides for your intelligence and your problem solving ability. In the rest of the body, these same networks are used to operate a different kind of collective intelligence, that of a bunch of cells, under which some of them which are neurons, but most of which are not, undergoing these kind of building projects, constructing and repairing the rest of your body. That's bioelectricity.
And when you say electricity and how it pertains inside the body, bioelectricity, um, are we just simply talking about the exchange of different charged particles or ions across the membranes of cells or through gap junctions, or is there anything else in that picture of this word electricity that you're using?
Yeah. So there are many aspects of it actually, and different people focus on different sides of it. So, for example, so everything begins with ions being unevenly distributed across membranes. So there's more of potassium chloride, sodium, and these kinds of things on one end or the other. Um, And as a result, there will be a voltage gradient across the membrane. So this is the kind of thing that we focus on in my group. There will be long range electric fields that other people study. For example, transepithelial potentials. There will be somewhat more exotic things like moving of electrons, which people like Frankie Dawson study. There will be all kinds of there will be static charges. There will be there will be other phenomena. So different people. There will be ion fluxes. So for example, Rich Nuccitelli and Lionel Jaffe made this vibrating probe that studies ion fluxes at a particular location. So for us, we put most of our effort in the spatial patterns of resting potentials. So this is the voltage gradient across the membrane. This is also what neuroscientists study in neurons, that right. So the voltage of a particular neuron and how it propagates to other voltages. Although people like Earl Miller are now really starting to get a lot of mileage out of looking at the electric field of that process and so on. But we study mostly the voltage patterns.
Okay. And so just for those who are not, in this world or, in healthcare and neuroscience, just to kind of explain it in a way where people like I can understand, we're talking about the difference between charges on one side of a cell membrane and the other is this voltage, this membrane voltage, and that's primarily what you guys are looking at and what we're talking about when we talk about bioelectricity and you guys, applying bioelectric signals in your work. Is that correct?
Yeah, so I want to emphasize a couple of things. So yes, it's the voltage gradient. It's basically a little battery, right? That's what happens in a battery too, is that you have a separation of ions, and the fact that they want to get together is what provides the motive force of that battery.
Okay.
So the thing is that really critical to note, and this gets back to your kind of the first question you had asked, which is this is fundamentally not about applying bioelectricity at all. So we do not do any field applications. We have no electrodes. There are no electromagnetic fields. There are no electromagnetic radiations. We don't apply anything, and that's because bioelectricity has a really unique aspect to it, which is that it is not just another piece of physics that you need to understand to know what cells are doing and why. It is also literally it is the medium in which the collective intelligence of body cells and also of your brain works itself out. So it is a natural endogenous way for the cells to talk to each other. This is the only reason we can do anything with bioelectricity is not because we apply external forces. It's only because that is what the organism is already doing, and we're just hijacking an existing interface. So cells already talk to each other through these electrical voltages, and they store information in patterns of these voltages, and they make decisions via the dynamics of these voltages. And it's only because all of that is true that we are able to tell these cellular collectives what to do by inserting messages into their native communication mode. And so this is really, really important. Bioelectricity has this a unique property. Let's think back thousands of years ago. People were able to train dogs and horses, knowing absolutely no neuroscience. They knew nothing about what's in their ears and between, sort of the two sides of the head. They knew nothing what's in there, and yet they were able to communicate with these creatures and to get them to do various things. And so this tells you a very important lesson, which means that you don't always have to exert control from the bottom up. So the modern paradigm largely focuses on bottom-up control. So people are very interested in the hardware. So protein engineering, rewiring molecular pathways, genomic editing, CRISPR, all of these kinds of things. Those are the things. Those are bottom-up controls of the actual hardware. And this is how computer science did its thing back in the 40s and 50s. Right? You can all imagine this photograph that I show sometimes in my talk of what it meant to be to reprogram a computer back then. So you would be sitting there and literally plugging wires in and out, right? This is where most of modern molecular medicine is focused. But we know that certain systems, for example, dogs and horses, and it turns out most of life offers an amazing interface that hides all that complexity. That interface is learning, and it hides all that complexity. So you don't need to know what is really underneath. But what you do need to know is how to send the right signals, so rewards, punishments, behavior shaping signals that are going to take advantage of the competency and the intelligence of the system to do various things. So that learning, all that whole interface is implemented by bioelectricity, of course, in the brain. But again, that architecture is not unique to the brain. It's not unique to behavior. The whole body works this way. And so that is why bioelectricity has a certain magic to it, which is that, yes, of course, it's physics, but it's a very particular assemblage of physics that provides an interface between high level information processing and the molecular stuff underneath that has to make it happen. This offers an unprecedented opportunity for regenerative medicine, for example, is to be able to communicate to that is to be able to communicate those various morphogenetic and other and physiological goals to that system without trying to micromanage it bottom up. And just just one last thing I'll say about it, which is that if I came on here and told you that just by the power of my mind alone, I can control I can depolarize 30% of my body cells. So most people would think that's crazy, and that's kind of an outlandish claim. But just keep in mind, this is how you get up out of bed in the morning, right? You have this high level cognitive intent that you're going to go to work, and in order for your body to actually stand up and walk, that high level cognitive intent has to filter down to the depolarization of your muscle cells so that you actually get up and walk.
Mm.
And so this kind of mind body connection and this idea that high level information like goal states and preferences and everything else feeds into and controls the molecular events in your body. It literally controls the chemistry of your body. That is not some weird, rare aspect of mind body medicine or meditation or a nocebo or placebo effects or any of that. It is something that the human architecture, whether the in all systems, the body architecture is doing 24/7. It's that's just how we work. So bioelectricity is this amazing layer that transduces between cognitive types of structures, so goals, memories, and so on, and the molecular hardware in your cells. So that's why I call it, I sometimes refer to it as software, not because it has any relationship to the particular kind of computer software that we deal with or the computer architectures that we deal with, but just the fact that that relationship between the hardware and the information that makes the hardware do different things in the world. And bioelectricity is critical to that.
Very good. And that, what you just said kind of makes me want to go off into more esoteric mind body connection stuff, but I'm
Yeah.
going to avoid the desire there. And I'd love to dig into and just get a quick summary of kind of three of the different experiments that you've done, that kind of also formulate my follow-up questions, and I think is going to continue to set a good foundation for folks listening. And that's the tadpole Picasso face experiment, the amputated frog leg regeneration, and then what you've done with planarians. If you could give a quick summary of each of those, it'll be a good launching point for some follow-up questions.
Sure. So the thing with the Picasso tadpoles is an experiment that talks about the importance of committing to experimentation as a way to assess the intelligence of unconventional systems. So people are often used to making these sorts of armchair decisions about whether or not things are cognitive, intelligent, whatever. And you think that by looking at something, you sort of assume you know what it is. And the framework that I'm working on, which is called TAME, stands for Technological Approach to Mind Everywhere, is one of the main tenets is that there's actually a lot of surprises, and we're not very good at recognizing intelligence and unconventional embodiments. And so we shouldn't be making these pronouncements. We should do experiments. We should make specific hypotheses and do experiments. And so here's one example of this. The tadpole of the frog has a particular facial structure, and then it has to metamorphose into a frog, which with a different facial structure. And so, in order for that to happen, all the components of the face have to move. So the jaws have to move, the eyes have to move, the nostrils have to move, all of this stuff has to move around. And it used to be thought that because every tadpole looks the same and every frog looks the same, all the pieces have to do is know how far to move in a particular direction, and it can be hardwired. You just do that, and you end up with a normal frog. So you look at that and you say, okay, that could be a hardwired process. The intelligence, aka problem solving ability there is very low, if not zero. So we decided to test that hypothesis because we suspected that there'd be much more intelligence to it than that. And so what we did was to scramble the initial organs of the tadpole face. So the jaws are off to the side, the eyes kind of on top of the head, everything is kind of moved around. And then we observe what happens. And those animals make largely pretty normal frogs because all of these pieces move in novel paths. Sometimes they go too far and actually have to come back a little bit, but they all move around in these in novel paths. until you get to a correct frog face, and then they stop. And so that tells you a number of interesting things. It tells you, number one, that this thing actually has a reasonable problem solving competency. That is, if you start them in the wrong positions, it doesn't just do the same thing and end up with a Picasso frog face. It actually has the ability to take novel action to reach the same goal. Now, this is the definition of intelligence by William James. It's the ability to reach the same goal by different means. So it has some ability to navigate that morphospace. We're back to this idea of shapes being locations in morphospace, and to change shape you have to move, you have to move the configuration in morphospace. So it has the ability to reach its goal from different starting positions. So that's pretty cool. You also immediately come across this idea of, in order to know when to stop, it has to know what a correct frog face looks like. How does it know what a correct frog face looks like? And this is related to something called the electric face, which was discovered and studied by Danny Adams in my group, who used voltage sensitive fluorescent dyes to look at the pre-patterns that enable the components of the face to know where they should go. She was using these fluorescent dyes to look at the pre-patterns that tell the pieces, the components of the face where they should be formed. And so this takes us right to the Planarian story, because there we asked a very specific question. We wanted to kind of do in the body what neuroscientists try to do in the brain, which is neural decoding. The idea is that if they could read out the electrical activity in your brain, they should be able to interpret that and read out your memories, your thoughts, your preferences, and so on. And so we wanted to know, where is this final pattern stored? This target morphology, is it represented, and if so, where? So we used planaria. Planaria are these amazing little flatworms that have a head and a tail and a bunch of different organs in the middle. And one of the things about planaria is, if you cut them into pieces, each piece regrows everything that's missing, and then it stops. So it makes a perfect little worm. The record is something like 275 pieces, I think. And so you can cut them into pieces. And so we asked the simple question: how do they know how many heads they're supposed to have? And there was already some indication back from work in the 40s and 50s by these guys, Marsh and Beams. And there was some indication that this was in some sense electrical. And so we used our dyes, and we found out that there's a bioelectrical pattern, meaning a spatial distribution of voltage potentials across the tissue, that encodes how many heads the worm is supposed to have. And what we found is that if we were to disturb that pattern, and the way you disturb that pattern is by using, in this case, is by using drugs to open and close various ion channels, which change the voltage of the cells. And so you can change the pattern. You can disturb that pattern, and you can rewrite it. Instead of one head, you can make it stay two. And so two heads. And if you do that, then you cut the animal, and they will go ahead and they build two heads. And several amazing things about that. One is that this much like some other experiments, tell you that these bioelectrical pattern memories are instructive, meaning they're not just epiphenomena, they actually tell the cells what to do. Number two, it's a kind of precursor to this amazing thing that our brains do, which is mental time travel, the ability to remember and to imagine things that are not true right now. Because this pattern, we can make worms that have this pattern that are not injured, that have one head and one tail, and yet if you look, their representation of what they should be building says two heads. So that pattern, that memory is latent, and it doesn't do anything until you injure the worm, at which case it becomes recalled and the worm builds a two-headed body. So a single-headed, anatomically normal animal can store at least one of two different perceptions of what a Planarian, representations is better, of what a Planarian should look like. And so that's kind of cool. It's a counterfactual memory. It's a memory of what you're going to do in the future if you get injured, right? And so this is neat, because you're literally, I told you at the beginning that I thought that bioelectrics was the medium of the collective intelligence of cells during morphogenesis. So now we're literally being able to read and write memories into this medium, the way that some neuroscientists can do in the brain. And the final thing that's amazing is that this literally, I keep calling it a memory, because it is. If you take a two-headed flatworm, you will cut it into pieces, and you will once again get two-headed animals, even though the genetics is untouched. So we never modify the genome. We don't do anything with CRISPR. Nothing wrong with the genetics of it. In perpetuity, as far as we can tell, forever, they will keep making two-headed worms. And so the question of what determines how many heads a Planarian is supposed to have is kind of subtle. On the one hand, the genome is relevant, because the genome builds hardware that by default always has a memory of one head. And so, but it's not hardwired, but it's not really nailed down by the genome, because by experience in its life, for example, the kind of signals that we give it, it can be moved to a two-headed memory, and after that, it's two-headed, even though the genomics is normal. So this brings you back to this idea of hardware and software, because of course, the two places that we know about things like this, where the hardware and the software diverge, is computer science and neuroscience. So in computer science, it's very clear that you can have hardware, and of course, the hardware is important for whatever you're going to do. But the exact same hardware is reprogrammable. It can do multiple different things. Even though it does one thing by default out of the box, you turn on the juice, your reprogrammable calculator always starts out saying 0, but you know that that's not the only thing it can do. And so this is what we found in the Planarian. And sorry, what was the third? There was a third example
The
you want?
the frog leg one, but
Ah, the
I'd
frog leg.
like to
The
say
frog
that
leg.
just it blows me away, even though I've heard it a few times. Just the idea that the anatomy that is regenerated, or if it's not if it's just kind of coming from the beginning, that it's not in fact, as so many of us probably have believed all our life, totally dictated by the DNA, the blueprint, if you will, of the body, that it can be completely changed by bioelectric signals.
Yeah.
It's really, I mean, the implications are so exciting.
Yeah. Yeah. No, it is. I mean, I think, and many people do think it this way about it this way, but it's been clear for a really long time that DNA is actually not a blueprint for the body. Even aside from anything having to do with bioelectricity, we know what genomes encode. They encode proteins. They encode the tiniest micro level hardware of every cell. There's nothing that you can read out of the genome directly that says anything about your body's shape or how many eyes you're going to have. This is not directly in the genome. So what we do need to do is to understand how that hardware is storing information and making decisions, and now we're starting to use that interface. Whereas in computer science, we stopped using a soldering iron and started using keyboards and monitors and things like this as a more convenient interface to take advantage of the competencies of the hardware, the memory, the reprogrammability, all of this. Bioelectricity, so evolution found this trick long before we did, of course, and is we are now starting to be able to do it. I should point out that it's not just about the number of heads. We've also shown that using very similar techniques, you can cause a planarian to grow heads that are appropriate to the species to the head of a different species, 100 to 150 million years distant. So
Yeah.
just imagine that morphospace, that space of possible shapes, has some attractors in it corresponding to different shaped heads, and there are species that live in those attractors by default, but you can get a different species to visit those attractors by changing the way it navigates that morphospace. And how does it make decisions where it goes? That's bioelectrical. So by controlling that bioelectrical decision-making process, you can get the same cellular hardware to visit a completely different region of morphospace. And so,
And so we'll come back to the frog leg, but I'd like to kind of take what you just spoke to with the planarians and the Picasso tadpole face, and kind of relate it to neuron regeneration. And one of the major challenges seemingly is, especially with like the motor neurons and how long the axons are. And if you're trying to regenerate this long axon, this idea of like going from the spine all the way down to the foot, it knowing the path to take and where to innervate and all that. Do you foresee there any connection or possibilities of this intelligence that we see, let's say in the Picasso face of it, you moving things around and it still knows how to get there through a different
yeah,
path,
yeah.
with this big challenging, regenerative path that a long motor axon would have to take? Any applications or possibilities with what you're doing applied to neuron regeneration?
Yeah, so we have a few applications that are specific, that kind of touch on that specifically. So we've looked at eyes transplanted to the tail of frogs, enable those animals to see, and so we have some ideas about how neurons know whether they're not in the right location or not. And we've looked at tail regeneration, which involves spinal cord and quite a long run of axons all the way to the tip of the end. But I think kind of more broadly conceptually, I'd like to bring up a different example, which is deer. So deer are a large adult mammal that every year regenerate their antlers. And the antlers are not like horns; they're real bone, and so they grow up to 1.5 centimeters of new bone per day. It's an unbelievable rate of bone growth. So they make bone, vasculature, innervation, and skin, the velvet that surrounds them. So all of this is rebuilt every year in this mammal. And so one of the cool things that this guy Bubenik discovered is that they have trophic memory, which is this thing where if you make a damage on somewhere on the antler in 1 year, the whole thing falls off. But the next year, the new tines that grow, there will be an ectopic branch point at the region where you damaged last year. So the plasticity, right? So the reason I'm bringing up this story, and by the way, I have these antlers in my lab. He donated when he retired. He donated because I was one of the few people that was citing his papers. He sent me 13 boxes of these antlers labeled, from a year, multi-year. Just think about that data set, right? Like 35 years of this year, and then looking at the shed casts and so on. So
Just to be clear, you get like a ding and a cut in the antler, and it sheds that, and the next year it's going to come back. It's going to show that cut still, that
it's
same
it's
dam.
going to show an ectopic. There's going to be an ectopic branch point, an ectopic growth. So
Oh, okay.
see, so the amazing thing about it is that, and I sometimes when I talk to my students, who have just had a course in molecular genetics and developmental biology and all that, I say, okay, here's this example. Draw me a pathway that might work for this. You've got the cells at the scalp who for months have to remember the fact that, let's see, 3 steps forward. And oh yes, don't forget an extra because we got damaged last year. But those cells are gone. That whole antler rack is gone. What's left are the cells underneath the scalp. They have to remember this three-dimensional pattern and changes there too. They have to take experience, this injury experience. I mean, what kind of a molecular pathway would even begin to explain this, right? Well, part of the issue is, and this gets back to the kind of what you said at the beginning about different disciplines and different and silos and all of that. It's there are different tools and different ways of thinking about it that are appropriate for certain problems. And of course, those are the problems that end up in the textbooks. It's the low-hanging fruit that's amenable to that kind of, so you have this beautiful developmental biology textbook, and it's full of examples that are properly analyzed by the molecular genetics of gradients and this kind of thing. But what's missing there, and this is what, when I talk to developmental biology students, I always start with the blank space, all the things that are not in your textbook. And the reason they're not in the textbook is because they don't fit well into the standard way we have of thinking about things. And so we talk about all the examples that are just not there. And so this is one of them. So the reason I bring all this up is because of the plasticity. So I'm not a neurosurgeon. I don't work on human length neurons from the spine down to the foot and all that. But I think that we have enough examples in vertebrates and including mammals like deer, where we can say the plasticity is probably much more than we think there is. And if we understood how to trigger that kind of anatomical homeostasis, the ability of cells to correct back to, correct for errors and do error minimization, I think a lot of these things would become possible. So, I don't have a mechanistic story to tell you about how to regrow a long nerve, unfortunately, but I do think the plasticity is way bigger than we give it credit for.
And just continuing on that idea, I know, like with the frog limb or any of the limb regeneration stuff that you've done, you guys have it involves a bioreactor. And I was kind of, something that, and you can explain what that means and what that is. But I'm wondering, is any of the stuff that you guys are working on in terms of causing something to regenerate not using a bioreactor such that it could be applied internally rather than just say like at the stump of an
Yeah,
amputation? But if we're looking at neuron regeneration, let's
sure.
say a spinal cord injury that doesn't have a long axon to regrow, but maybe a short area of trauma, can things be, or have things been applied internally, and you've seen these regenerative potentials kind of manifest in any kind of scenario like that?
Yeah, yeah, absolutely. And so I can tell you two stories about that. The thing about the biodome is that if you're thinking about cells regenerating a limb, let's say in a mammal or something else that's out in the facing the outside world, it's really critical to have a protective covering to convince these cells that it's safe to regenerate, that they can exert effort to do so, and to protect them from drying out and from give them this almost an amniotic like environment. So the biodome does a nice job on that for outward facing appendages. But here are two stories where we didn't use a biodome. So one has to do with tail regeneration. So tail it has muscle and spinal cord and all of that. And so we did a project. This is goes back to Kelly Chang in 2010 and Danny Adams before that in 2007, which is to take tadpoles at stages where they normally do not regenerate their tails and apply a drug that puts the regeneration blastema into a specific bioelectrical state that triggers regeneration. So when you do this, you don't need a biodome. You soak the whole animal. The amazing thing is that nothing else in the animal goes wrong. It's only the cells that are at the wound that seem to pay attention to this. So that was kind of neat. And what it does is it triggers regrowth of the complete appendage. And so in Kelly's, specifically in Kelly's work, we had 1 hour of treatment. That's it, just 1 hour of treatment, and that gave rise to about 10 days of tail growth. And so this matches well with our whole strategy for regeneration, which is that we're not looking to micromanage what cells do during this process. We're not trying to tell every stem cell what to be or what all the gene expression should be. I mean, that's not going to be feasible in our lifetime to micromanage it that way. But what we are trying to do is to give it a high level signal at the very beginning that convinces the system to head down the regeneration path instead of the scarring path. And so, in the case of the tail, 1 hour soak, don't touch it again. 10 days, full leg regeneration, and then
And I want to make sure I heard you correctly. You said that basically the entire tadpole was exposed to this drug, but only the cells that you or the area that you wanted to spark regeneration were actually affected or responded to that drug. Did I hear that correctly?
that's correct. Yeah, and we've done this. So we did it with the tail. We did it again with the frog leg, where the same kind of approach was done with froglets to soak the whole animal and trigger leg regeneration. And nothing else that we could notice went wrong. The same thing happened in our work on this is Doug Blackiston's work on transplanting eyes to the tail, and they start to grow these. We can induce with a drug them to grow these ectopic optic nerves, but we just soak the whole animal, and the nerves in the head ignore this completely. And so I think that well, I think what happens is that the cells are in the correct position and know it, are not paying attention to these cues. What the only thing paying attention to these cues are cells that can tell that something's wrong and they don't know where they are. And so they need to figure out what they should be doing. So either because there's a wound or their stress levels are high for whatever reason. I think that, and then I'll tell you another story which doesn't use a biodome, which is repair of brain function in birth defects. So this is the work of Vaibhav Pai in our group, and he collaborated with Alexis Paitak and some other people on this. Uh, Basically, what we did was to look at the early embryonic brain and look at what happens to the bioelectric pre-pattern that tells the brain what size and shape it should be when you expose those animals to a bunch of teratogens. So alcohol, nicotine, various other toxins, or mutations. In fact, even mutations of really crucial genes like Notch, really important neurogenesis gene. So turns out that when you do all those things, the pre-pattern for the brain goes wonky, and then the brain is misformed. And so these animals have no behavior, the brain is misshapen. It's a terrible, really, really profound birth defect. So Alexis was able to make a model where we asked, okay, given that we know what the correct pre-pattern should look like, we know what the incorrect pattern looks like, what channels could we open or close to get the wrong pattern to look like the correct pattern? And the model gave us a suggestion and said the HCN2 is a particular kind of channel that could do this. And sure enough, when we soaked those animals in the drug, no biodome, whole body soaked, we got brains that were indistinguishable from controls, both in terms of shape, gene expression, and IQ. They got their IQs back, which was just, and so a couple of amazing things about this, right? One is that we've gone from very early work using screens to just ask what's possible when you change the bioelectric state of the body, and we discovered you can make new eyes and you can do all sorts of things, down to the point where we now have a computational platform which can tell us exactly which drug to use to cure a very complex defect. We don't have any hope of micromanaging the shape and size of a brain, even a frog brain, it's too complex. But we don't need to, because there's a nice simple bioelectrical pattern that tells the cells what to do, and there are ways to compute what channels could be tweaked to sharpen up that pattern where it's normally screwed up. And the other most amazing thing to me of all here is that one of the things that we rescued this way was the Notch mutation. And so the Notch mutation normally wrecks, it's an overactive dominant Notch, it just wrecks the brain. You can fix this fundamentally hardware defect. So the Notch signaling machinery is broken, you can fix this defect not by re-editing the genome, not by fixing the notch, but by forcing the correct bioelectrical pattern in software. I say software because we're just providing a soak in this drug. We're not rewiring the genetics. And so I'm to be clear, I'm not saying that all genetic problems are going to be solvable this way. There will be many things like metabolic disorders where someone's just missing an enzyme. You know that bioelectricity is probably not going to help with that. But many of these disorders are probably solvable. Even genetic disorders are probably solvable in software if we understood the right way to interface to the tissue and prompt better responses than what it naturally does. So those are two stories that don't involve biodome.
Well, that just brought up a bunch of things, my kid's having an actual genetic, a single gene,
Mm,
issue.
yep.
And they've gotten a gene therapy, but
Mm,
it's not really panned out to have the same effect on everyone that everyone was hoping for,
mm,
although still miraculous. But when you say this, if we can figure out this interface of how to make that communication to, say, all cells, how is it different than saying, like just bathing the entire tadpole or bathing the entire frog with this drug or this set of drugs to create the voltages you want? I mean, it almost sounds like you kind of did. It's like, the cells that are good to go and don't need the signal, they ignore it, and those that do need the signal, they don't. So,
yeah.
we're, um, please
Yeah.
explain further there, and maybe also like when will we see some of this in humans? Some of
Yeah.
this work.
Yeah. Well, let's see. I mean, keep in mind, right, that the examples that I'm telling you about are the things that worked first. So these are the things that have already worked. So these are the easiest, obviously, these are the easiest examples. So there are going to be many things that cannot that are going to need a more nuanced approach, right? So we found some of the things that for example, in the case of the brain, what happens to this bioelectric pattern is that it sort of becomes fuzzy. So the difference between so here's the edge of the brain, and here's some cells that are not going to be brain. And normally there's a strong voltage difference, and that makes a nice sharp line, a nice demarcation of the brain. And if that pattern gets kind of fuzzy, then the cells don't really know where the borders of the brain are, and it becomes a mess. What bathing it in that drug does is it opens up this particular channel, HCN2. What it does is any cell that's depolarized that leaves it depolarized, any cell that's a little bit hyperpolarized, it jacks it up and makes it more. And so it sharpens that. It's like a sharpen filter in Photoshop. It like just right. And so that's very convenient because then you can bathe the whole animal and you don't need to worry about where it's going and all that. So I'm not claiming that everything is going to be solvable like this. That'd be nice, and I think many things will be solvable like this. But ultimately, we're just scratching the surface of this bioelectric code where the temporality of pulsing, and remember, we just put the drug in, we don't even like pulse it, we just put it in and leave it there. Uh, the temporality of how you stimulate, the spatiality of how you stimulate may turn out to be really important. Um, it's not as bad as you might think. I mean, originally everybody said, "Oh my God, you're going to have to tell every cell what voltage to be," and obviously that's not true. The other amazing thing that the more recent paper was we took that approach to fixing the brain and we said, well, can we use it to fix heart and gut defects? The problem with heart and gut is that you can't see them. The brain is kind of right up at the top, so it's pretty transparent at the stages that we need to see. You can see what's going on with in terms of the voltage. The heart and the gut you can't. It's underneath a lot of tissue layers. We said, how are we going to do this? We can't see what the correct pattern is, and we used just exactly the same technique basically without seeing anything of what the natural pattern is, and it works. It fixes in the frog model, it fixes gut and heart and I think face defects as well. So I think there's a long way to go with these approaches where you can do this in a way that does not require you to understand everything that you need to about the bioelectric code. But ultimately, we're going to need to crack this code. There's only going to be so many things that we get lucky enough with. Where there's a simple, ubiquitous trigger that doesn't need to be modulated in any way that tells the cells what to do. There will be some of that, and then there will be some more complex things that we really want to. I think that if you're going to want, for example, if you're going to want to for synthetic biology purposes for biorobotics, you're going to want to make shapes that don't exist in the real world. You're going to make other we're going to want to make other kinds of constructs, and I think to really gain control over morphology we're going to have to crack this code completely.
And when you say crack the code, you're saying like the voltage map for each
Yeah.
particular tissue type, organ type, whatever the case may be.
We have to go beyond that because that will tell you how to make organs that already exist in the body, and that's great for biomedicine. I mean, that's amazing. That would be amazing. But I want to go beyond that. I want to be able to compose new sentences in this code. I don't just want to use words that, it's sort of the difference between learning a language at the beginning. You learn a foreign language and you sort of know, apple, car or whatever.
Mhm.
Uh, but what you really want is the ability to compose those things into novel ideas that are have not been said before. It's not something that you can just point to and see that, here's this thing I want that already exists. So ultimately, we want to understand how cells and tissues interpret these electrical patterns. That's the name of the game, right? It's about communication. And that means in order to communicate effectively, you have to know how your target is interpreting the things you're saying. So it's a matter of figuring out how the interpretation works.
Mhm. And back to a question I asked just a little bit ago, I can imagine that people interested in spinal cord injury, their ears are perked up. And when you were talking about, these mutations in the brain and it just coming back and even intelligence, that those who are interested in traumatic brain injury, I'm sure are quite interested listening to this. So, what would you say is the path um, to getting to testing and experimenting and doing some studies in humans?
Yeah.
How long do you feel that path is?
Yeah, everybody asks about the timeline, understandably so. I would be lying if I gave you a specific number of years because I don't think we can know that. This is science, and it depends on how the discoveries go. It depends on funding. It depends on many things that are not predictable at this point. But the roadmap is roughly going to look like this. We need advances in physiomics, so we need to be able to gather these data sets. We need to know what the correct bioelectrical pattern looks like for health, disease, different ages, and so on. We haven't even talked about the cancer, the whole cancer thing, because there's a huge role of this in cancer. And then we need to have the computational tools level up, and then we need to have the delivery technology. So next generation of biodomes, the next generation of pulsed drug application devices, and so on. All of that is being worked on now, and I have to do a disclosure because David Kaplan and I are co-founders of a company called Morphoceuticals. So we're doing all of this stuff as part of Morphoceuticals. So there's
Mhm.
my conflict of interest disclosure. But the next step is mammals. So we've done all this stuff in frogs. We are now in mammals. So we're doing experiments in appendage regeneration in mammals and cancer in bioelectrics of cancer in human cells and so on. And so mice and things like that, and then larger animal models, and I hope I hope soon humans. I can't give you a precise timeline, but we have a company around this. We are planning talks with the FDA. I expect to see this stuff in my lifetime. I hope, but I can't give you a specific number of years.
Okay, no problem. And I know you have to run, so we'll go ahead and wrap it up there. Hopefully, we can do a part two,
Sure, happy to.
continue the conversation.
Yeah, happy to.
Thank you so much. What would be like a good website for those who want, are interested in learning more about your work and
Yeah.
really digging in? What's the best place for them to go?
Yeah, absolutely. Everything is at www.drmichaellevin.org. One word, drmichaellevin.org. And everything, the papers, the talks, the data sets, software, everything's there.
All right. Well, thank you so much for your time and for this incredible work. I'm so excited to see where it goes and hope I can help you guys to push it forward.
Thank
And
you so
yeah,
much. Thank you. Yeah, thank
we'll
you.
see you in part two.
Yeah. Thanks for the conversation. Really appreciate it. Best of luck to you and your kids. And yeah, I'm happy to do a part two anytime.
Awesome.